Okay, this lecture is some basics on immunology. I apologize if it's a little long, but I'm going to try to just go straight through it, so pause whenever you get tired of listening to me. We're not going into all the details of immunology. There's a separate class for that, but I want you to know some of the terminology. We'll talk about those in future papers that we read as a class, and so this is kind of a background on how our body responds to pathogens. So most of the time when an organism infects the body, the immune system is able to prevent replication and spread of the disease. Okay? But not always. And so what's happening is when you have an infection, okay, you have two different types of immunity that come into play, and they come into play sequentially. So the first one is your innate immunity. Innate immunity is nonspecific. It's short-lived, and it's your first response. And many times, your innate immunity can take care of the infection, and you're done, no disease. Sometimes the innate immunity does not clear the infection, and you start to show disease. And this disease can just be symptoms like a runny nose and a fever and a cough. And what's happening in the meantime is your second kind of line of defense, your adaptive immunity, is now kicking in. Okay? And what's different about the adaptive immunity is that it leads to memory. The next lecture you'll watch is about vaccines. And vaccines are all about eliciting this adaptive immune response, or this memory. Adaptive immune response is also specific, and we call it cell-mediated because it involves B and T cells. So let's look at these different types of immune response independently. So, in order for your innate immune response to kick in, first the microbe has to get into the body, right? And you know we've talked about some of the physical, chemical, mechanical barriers that we have. We have our skin, we have mucus, we have tears, we have enzymes, we have mechanical movement, sneezing, coughing, all of those things to try to prevent an infection. But if the pathogen is able to get into our body, then our internal system, which is our innate immune system, kicks in. And the parts of it we are going to talk about are phagocytosis, complement, interferon, and natural killer cells. Phagocytosis. So I highly suggest at some point you click on these movies. They're really fun to watch. They're showing you how your body goes about, or your cells go about eating foreign objects. Okay. So this one I call the big meal, and this one I call the chase. And you have two types of phagocytes, two types of cells that can eat. And you'll watch, when you watch the movies, you'll see it engulf, move around, take in. And then if you remember about your lysosomes, the digestive enzymes in your cell, these lysosomes fuse to this poor little bacteria or protozoa or whatever the macrophage has eaten and start digesting it. So macrophages are one of the main types of phagocytes, and neutrophils are the other type. Macrophages are much longer lived. Neutrophils have a shorter lifespan. What's really interesting about neutrophils is they actually don't have any mitochondria. So they just rely on glycolysis, and because they can live without mitochondria, they can go into anaerobic spaces, right, without oxygen, because if you're using cellular respiration, you need oxygen. So they can go into parts of our body that are anaerobic and phagocyte or eat up whatever foreign objects are there. We're going to primarily focus on phagocytes, on macrophages. And this is a figure from your book that is just showing that you have macrophages throughout your body. So they are going around surveying or patrolling, looking for foreign objects. They can be recruited by something we call cytokines. And cytokines are cell signaling molecules. And these are all blood cells, white blood cells. So these come from your bone marrow. And this is just to show where some of your sources are, where the macrophages are hanging out. So what do the macrophages do? Well, they recognize foreign objects. In this case, we're talking about pathogens. And this PAMP stands for pathogen associated molecular proteins. So what we think is that there are some proteins that are expressed on the outside of, say, a bacterial cell that the macrophages can recognize as foreign. And what happens is when the macrophages bind, they engulf. So they move their cytoplasm around that little bacterial cell. They fuse it with a lysosome, so phagolysosome, and then that results in digestion. Okay, so they bind, engulf, and digest, and spit out the waste. Right? So very efficient way to go through and kind of clean out things that are foreign. Now, obviously, not everything gets recognized, and there is a little bit of controversy as to what exactly the macrophages are recognizing, because some non-pathogenic bacteria also express these PAMP proteins. So if you take immunology, you will find that it is a very complicated system, and we are always learning new things about it. All right, so we got macrophages, or phagocytes, the things that come around, survey, eat up foreign stuff. Another part of the innate immune response is the complement system. And you are not required or even expected or even suggested to go through and memorize this. Again, details for this would come in an immunology course. What I want you to understand is that you have these proteins, and all these C-labeled proteins are complement proteins. And there's about 30-some proteins that make up the complement pathway. And when the complement pathway is activated, you get this cascade of enzyme activity. And in the next slide, we'll talk about some of the outcomes of this. Again, we're not going into these details, but just so you know, the classical complement pathway is triggered by antibodies, which we're going to talk about in a little bit, and the alternative pathway is triggered without antibodies. Okay, so let's look at a little, maybe more simplified idea of complement. So in this case, we have a little pathogen, a little microbe, okay, and some antibodies. We'll talk about antibodies in a minute if you're not sure what those are. Some antibodies bind, they recognize this microbe as foreign. The classical complement pathway is triggered, so it binds the antibodies, and then you have this whole cascade of events. Again, this can also be triggered without the production or presence of antibodies. The outcomes are the same. One outcome that we will talk about is opsonization. And you'll see the word phagocytosis. So opsonization is another way to tag something and say, hey, phagocytes, come eat this. Okay. If a microbe, particularly bacteria, are being targeted by a complement, you can get a MAC, membrane attack complex, which is basically, no, that's not the right, punching holes in the cell membrane. And you can imagine if you start punching holes in a cell's membrane, it's going to die. Okay. The other word up here I want you to see is chemotaxis. And that means using chemicals to attract other cells. Attract other cells. Okay. So complement has lots of different proteins involved, multiple ways to recognize something as foreign, and then multiple tools to use to recruit other cells of the immune system, to recruit phagocytes to eat up whatever it's tagged, or to punch holes in the cell membrane to kill that microbe. Another part of your innate immune response is interferon. And at first we're going to talk about interferon alpha and beta. Okay. So interferon is also called a cytokine, so a cell signaling molecule. This is an early response, and it's really important for virus infections. So what we have found is that when a cell is infected with a virus, these molecules called interferons are produced, and they go and tell the neighboring cells, hey, there's a virus around, you need to be ready, put up your defense, get ready to kill it. Okay? So this interferon is working on or signaling neighboring cells and let's go to the next slide. What happens is you actually turn on antiviral genes. So the cell, the neighboring cell, here's the one that's got the virus in it. Here's the other one, and it says, hey, turn on some genes so that if you get infected by a virus, you are ready to shut it down. So there's a couple genes that turn on that help inhibit expression of viral RNA. Another thing that interferons do is they increase in expression of MHC. And all I want you to know about MHC is basically it's a signal to tell cells to tell other cells I'm infected. Okay. So this infected cell could produce MHC and say, help, I'm infected, come kill me. Right? Table 14.5 in your textbook is a good summary of these interferons and their actions. Because in addition to interferon alpha and beta, and remember, I want to step back. Remember that this is part of the innate immune response, so it's non-specific. So this is not specific to any certain virus. This is to just viruses or pathogens in general. Okay. Interferon gamma is actually part of the adaptive immune response. And so I'm just bringing it up here because it seems to flow good with talking about interferon. And what I want you to see is that it's made by natural killer cells, which we're going to talk about next, and T cells, which are part of the adaptive immune response. And then it stimulates all these other cells. So it gets them activated. It says, all right, we've got an infection going on. Hey, macrophages, we need you to come and chew things up. Natural killer cells, we need you to come. We need our B cells and T cells. So it helps recruit the immune system to the site of the infection. Okay. Natural killer cells are also part of the innate immune response. And what they do is they circulate around, and they're kind of a double check to make sure cells are not infected. So, MHC class 1 is a way to say, I'm good. Okay. So when the natural killer cell checks out this little other cell, it says, oh, you're a good cell, I'm going to leave you alone. When that MHC class 1 is not expressed, okay, it's not saying, hey, I'm self, leave me alone, then the natural killer cell says, it's time for you to die. Okay? And it produces all of these granules, which are basically proteins. And these proteins put holes in the membrane and allow other enzymes into the target cell. And the final, I just lost what I was going to say. Apoptosis is what I'm trying to say. I can't figure out how to connect over there. But the end result is apoptosis. So apoptosis is programmed cell death. So what's basically happening is the natural killer cells are going around surveying your cells. And it says, okay, you have MHC class 1, I'm good. Great. Oh, you're not expressing self. You must be infected. I'm going to release these enzymes. They're going to go in. They're going to trigger apoptosis. You are going to die. So those are what are natural killer. Again, it's not specific. This guy doesn't know what the target cell is infected with. It just knows it's not saying, I'm good. So here's a image of a natural killer cell. and a target cell interacting, and I just think it's a really cool picture, so I shared that with you. One other type of cell that I think is pretty interesting are the eosinophils. This is a type of white blood cell, and eosinophils target large parasites or pathogens. Okay. So they have this G is all these granules, and granules just mean sacks of proteins. Okay. So it is floating around, waiting and waiting and waiting to detect something, and it can detect something like a helminth. Right, so here's a little worm. This worm has been coated with C3b, which is part of the complement. So the complement recognized it as foreign, attached the proteins to it, oops, sorry, and then all these little eosinophils attached to the complement. And so now this poor little worm is coated with white blood cells. The eosinophils release all these proteins, and they basically give the parasite what we call a chemical burn. So they release reactive oxygen species and cationic proteins, and this just damages the pathogen's membrane. Okay. So what's happening with something that your macrophages, now can you imagine this little guy, if he was the size of a macrophage, could not eat this big worm, right? So instead something large, is coated with complement. The eosinophils recognize the complement, they attach, they release all these proteins. The proteins damage the pathogen's membrane. Pathogen dies. So those are examples of our innate immune system. They're not specific to any specific bacteria or virus or even worm. They're just serving for foreign objects in general. Now we're going to switch over to the adaptive immune response. So humoral is talking about antibodies and B cells, and cellular is talking about T cells. Okay. So remember that adaptive means specific, and adaptive means memory. Okay. So your body has been working and working and trying to get out this pathogen, whatever's infecting you, and finally, and sometimes this takes a day or two to kick in, your adaptive immune response comes and says, all right, I'm going to clear this thing out. One of the most important components of the adaptive immune response is antibodies. So antibodies are proteins that are made by B cells, and they are very specific. Oh, I don't know, I think there must be something I just hit somewhere. Very specific. Okay. So hopefully you've heard of antibodies before. We use them in molecular biology. We use them to do lots of research, but they're also a huge part of our immune system. And they're very specific because these variable regions at the tips of our antibodies are specific to one type of antigen. Okay. And the cool thing is, hopefully you may have learned this in genetics, and there's information in your book about how these proteins are made. They're made by genetic recombination, random recombination of DNA. And so every time you produce a new antibody, you produce something slightly different at the tips. And when that antibody recognizes the antigen, that's the foreign object, then you will see the antibody, excuse me, stimulates the B cells to make more. It's pretty cool to think that this is totally random as far as what antibodies you make, and yet we make these very specific antibodies that will recognize pathogens and turn on that adaptive immune response. Okay, so some of the ways that antibodies work. Antibodies can work with complement and phagocytes for something called opsonization. So opsonization is basically the tagging of a pathogen and recruitment of phagocytes. So remember, with your complement, you can do it with or without antibodies, right? Phagocyte recognizes the complement, okay, binds and eats, destroys that, in this case, bacterium, the pathogen. Okay. So antibodies can take a role in this, and what this is trying to show you is that here's your innate just complement. Once you start putting antibody in there, and antibody with complement, you bring down the levels of bacteria, or you accelerate the phagocytosis by using these two pathways. Okay, antibodies can also trigger mast cells. So mast cells are another type of immune cell. And so in this case, the antibody is recognizing a microbe, or in this case also this little thing, I don't know why they drew it this way. This is supposed to be some kind of foreign object. Okay. And when you get a couple of these antibodies together and this mast cell, the mast cell releases, it degranulates, so it's basically releasing proteins. What's happening when mast cells release these proteins is you get inflammation. Okay? So inflammation is you increase what we call vascular permeability. So you get more fluid flowing. Okay. You are recruiting immune cells. Okay, so this is your chemotaxis. And all of this leads to inflammation. So here you go, chemotaxis, vascular permeability. This foreign object could also be what we call an allergen, right? So if you take an antihistamine, histidine, histamine. If you take an antihistamine, it blocks mast cell degranulation. You don't get a runny nose, you don't get recruitment of cells, you don't get inflammation, you don't get a bunch of mucus buildup. Okay. There's a lot of controversy, I don't know, discussion, I should say, as to whether it's really good for us to take cold medicine and things that relieve our symptoms when we're sick. Because what you're doing is you're blocking your immune response, right? Unfortunately, most of us have school and work and kids and places to be, and so we take things to make us feel better. But there are some studies that suggest that by doing that, you're actually prolonging your infection. So, just something to think about. Hey, you're taking an online class. You could be sneezy and coughy right now, right? And still getting your classwork done. So, awesome. Antibodies and phagocytosis. Antibodies can actually trigger phagocytosis without even going through the complement system. Just by their binding, they tell the phagocytes, hey, there's something foreign, come eat this up. Antibodies can block interactions. So when you get a good immune response, or actually when you get a vaccine, one of the things we try to do with a vaccine is make what we call neutralizing antibodies. Okay. That's your goal. When you're evaluating a vaccine for effectiveness, you're seeing, does it make antibodies that block the infection? So that's what a neutralizing antibody does, blocks the infection. Okay, so for instance, the antibody is binding to the little virus here. So it's not allowing the virus to bind the receptor and infect. Here, the antibody is blocking something on the bacteria so that it can't take up nutrients. So that's going to starve the bacteria to death. When you watch the vaccine video, you'll know that some of the vaccines we make are against toxins. So chemicals that bacteria or viruses, bacteria, produce that can cause us harm. So we have vaccines against toxins, and what they're doing is they're making these antibodies to bind the toxin so that the toxin can't harm our cells. And this diagram is showing that this antibody is binding to the infectious agent to prevent the infectious agent from getting into the tissue. So the mucosa, it's trying to cross the lining, get to the cells down here. That's a cell, that's a cell, that's a cell. And so antibodies can again block that interaction. Remember, antibodies are very specific. And that's the basis of how a vaccine works. If you get a pretend toxin to work on, and you make an antibody to that toxin, and we're going to talk about those antibodies become in memory. Now you've got a specific antibody for that toxin floating around so that in case you come in actual contact with it, your body is ready to mark it for destruction, block it from working. Okay. Antibodies can also mark large parasites. So here's like probably a little worm again. You've got your eosinophils, which we talked about, can also interact through complement. You've got your natural killer cells, part of that innate immunity. Macrophages, okay, he's probably not going to be able to engulf that, but they can all produce proteins that help destroy that parasite. So there are 5 different classes of antibodies: IgG, IgA, IgM, IgD, IgE. You do not need to know this table. I just want you to take a look at it and you can see some of the differences. So some work with complement. IgG is the one that actually crosses the placenta and helps protect your baby. Some work with macrophages. Some work with the mast cells. Okay. They all bind antigens. So they're all specific, but they're produced in response to different types of foreignness, allergies, bacteria, or they're located in different parts of your body. So, where do these antibodies come from? They're made from B cells. Okay, so in the B cell, in the B cell DNA, you get this rearrangement of the genetic material. It produces these new kinds of proteins. And when those antibodies specifically bind to the antigen or to the thing it recognizes, it signals the B cell to start dividing. And the B cell, will eventually make what we call plasma cells, and this is your memory. So it turns on this cell to say, hey, we really need a lot more of you. Divide, divide, divide. Make more antibody. Destroy that foreign object. Each type of B cell makes a specific antibody and makes a different type of antibody. And sometimes you need additional signals to get this memory cell. But the main difference between a plasma cell and this immature B cell is these are now secreting antibody. We call this soluble antibody. So now this antibody can go without being attached to a cell and float around and survey and find antigens. So one of the ways they can test to see if you've been infected with something specific is to take a little bit of take a little bit of your blood and test for antibodies. So do you have antibodies to Zika virus? Do you have antibodies to HIV? Do you have antibodies to influenza? Do you have antibodies to hepatitis? Because these are long-lasting and they're circulating, you can detect what people have been exposed to. Remember that these plasma cells are now going to be our memory cells, so they're going to continue on dividing, releasing antibody, and preparing us for that second attack or exposure. Okay, next type of cells I want to talk about, and we're almost done. Okay, this is going long, is the T cells. So B cells mature in the bone marrow, so they're called B cells. T cells mature in your thymus. Okay, derived from the same place, both part of the adaptive immune response. And you can see that we have different types of T cells, different functions. I just want to briefly talk about a couple of them. The Th1 or the T helper cells. Okay. These are really good for internal infections or internal pathogens. Okay, so you can see there's a parasite inside the macrophage. So this macrophage. So he can't know to eat up this stuff. This parasite is hiding out. So viruses do this, Listeria does this, Rickettsia, tuberculosis does this, and they hide out away from the immune system. Luckily, the T helper cells can come in and recognize this class 2. Remember, class 1 is self, class 2 is foreign, and say, oh, dude, you are infected, you need to kill that parasite. So here's interferon gamma we talked about, part of that adaptive immune system. And they activate the macrophage and say, you have a foreign invader, get rid of it. There's also a type of T cell called cytotoxic T cells. These are really important for virus infected cells. So here's your virus infected cell. It's producing an NHC class 1 molecule, but somehow it tells this guy, man, I'm infected. You need, oh, I'm sorry, that's how. Okay, back up. Start over. This is a virally, virally infected cell, and the cell that is infected is expressing or producing a virus peptide. Okay. The cytotoxic T cell, this T cell receptor, is highly specific. It's analogous to an antibody from the B cells. Okay, so this is also made by random DNA rearrangement. And so once this T cell recognizes this antigen and says, oh, this cell has a foreign pathogen in it, it activates the cytotoxic T cell. Cytotoxic T cell sends signals for that infected cell to self-destruct. Okay. So this is similar to your natural killer cells recognizing, but in this case, it's very specific, and these T cells can become the memory T cells, right? So you've got this specificity that says, hey, anytime, let me clear some of this up, anytime you see this molecule again, kill that cell. Okay, so that's where your memory comes in. So there are a lot of responses to virus infections, right? You've got your T cells, your natural killer cells, your interferon signaling, and this is important because remember, viruses are obligate intracellular parasites, which means they are hiding out in our cell. So it's not like you have another cell, a bacterium, or you have a big worm, helminth, in there to recognize as foreign. These viruses are inside your cells. So your cells have evolved ways to tell the outside, hey, I'm infected, would you please kill me so that we don't spread this virus. So I've said memory, memory, memory. I want you to really understand that the adaptive immune response is about memory. So you can have B cells and you can have T cells as part of that, part of that memory. Clonal proliferation means once they get stimulated, they make more of themselves. Okay. This is important for that secondary response. This is your goal of a vaccine. Make memory, be ready, have those cytotoxic T cells, have those antibodies ready so that as soon as that pathogen comes in again, we're immediately ready to knock it down. Another picture of the adaptive immune response I'm putting up here is just to show you that different pathogens trigger different types of immune response. Okay, and One of the really interesting things is that an overactive immune response can also lead to disease. So you see harmful inflammation, right? And so we've talked a bit about using helminths to overcome that hygiene hypothesis, where we're so clean that we haven't got this good immune response to keep things at homeostasis. And so that when we see certain things, or when we're exposed to pathogens or foreign objects later in life, we get this overactive immune response and things like harmful inflammation. Autoimmune disorders is when your immune system is attacking yourself. Okay. So remember, have your kids eat dirt. It's good for them. All right, so we talked about innate and adaptive immune responses. They have different components. You'll see in the homework, I'm going to ask you to kind of do some summaries about this. Again, it's not for you to memorize, it's for you to get familiar with the terminology so that as we start reading papers, you know what they're talking about. All right, your last video for this week is on vaccines. Thanks.